So this week in lab, you'll be isolating DNA from your phage, and you will use your high titer phage lysate that we generated in lab last class, which means that when we work with this lysate, we want to make sure that we have a sterile environment. So you should be using the flame and sterilize your bench top just as we've been doing. But after we close this lysate back up and we have our 1 milliliter of that lysate collect into a tube, we no longer have to keep our flame on and we no longer have to stay sterile. So to start off, just like normal, we'll light our flame, have our sterilized bench top, and be able to work. We'll collect 1 ml of our phage lysate, and this time we're going to use a P1000 pipette. Most of us have around 9 to 10 milliliters of lysate that was collected from those 2 plates that were flooded. So we might need to share because we don't have enough P1000 pipettes for everyone, but you'll notice that it's P1000 on the side of pipette, and we will use these larger pipette tips that have been in your box all semester. So I already have a tube ready for adding this phage lysate to. Get myself a pipette tip, keep close to the flame, hold the lid in your hand, have this pipette set to 1000. We'll get 1 ml of that lysate and put that in to an empty Evansville tube that you have waiting. Close it up. Now we can turn off the flame because we actually don't need to stay sterile anymore. As long as the original tube of your high titer lysate is sealed, this will go back in the fridge for us to use at a different time, but we don't have to worry about staying sterile for the rest of this lab. So our goal today is to isolate DNA from this phage lysate that we put into a tube, and we hope to have about 5 times 10 to the 9th titer in order to collect DNA from this phage. Some of us may not have a titer that's quite so high, but hopefully we'll be able to still isolate enough DNA from that phage. So following protocol 9.1, we will start to extract DNA from our lysate. To do this, you should have some EDTA, a tube of the nuclease mix, and those are going to be provided for you. You should also have 2 of these columns that look like this, and 2 tubes that you get out for yourself that are new sterile Eppendorf tubes to put those columns in. You should also have 2 of these syringes. These 3 milliliter syringes, we're actually going to take out the plunger itself and have just the barrel available. And we're going to screw this barrel into the column, right? So that just screws on there. Do not screw this on too tight. You want it just to the point of resistance. If you screw this on too tight, it'll actually screw past it and come off more easily. So this should feel like it's on pretty snugly, but don't take it too far past that point of being snug. The last things that you should have are 2 tubes. One of them is going to be a DNA resin. You will notice that probably it's been sitting out and it has separated a bit where you have these beads. This is a polymer that is going to bind DNA. And because this has been sitting around a little bit, it may make sense to vortex this just to mix this up and have those beads resuspended. And the other tube that you will have is 80% isopropanol that will be used to wash the columns. So when working with the nuclease mix, it's good to wear gloves. So I'm going to get my gloves on, and you'll add 5 microliters of the nuclease mix to your phage lysate. Once you've done that, this will actually incubate at 37 degrees for 10 minutes using a heat block. So here's our tube and our heat block. Like I said, we'll incubate here at 37 degrees for 10 minutes. Once our 10 minutes is up, we will pipette 15 microliters of EDTA, which is a calcium chelator, into our lysate. It's important to mix a little bit at this point. So what you'll want to do is you want to take that lysate with your EDTA and nuclease mix and just give it a little bit of a flick. The next step is actually done already for you. The protocol says to put 2 milliliters of that resin into a tube. We already have 2 milliliters of resin that's pre-aliquoted for you, but it is important, like I said, to mix this because it is a slurry of these beads that are going to allow us to precipitate DNA, and so you'll want that to be well suspended. So now we're going to take our nuclease treated phage lysate and add this to the DNA resin in this tube. And since you have a little over a milliliter, you'll probably want to pipette this twice. And you can use the same pipette tip for this. So I'll get some of it, transfer that to the DNA resin. And then go back in, get the rest. Now you want to mix this by inverting. And so what you'll do is actually go back and forth like this continuously for about 2 minutes. So after we've done 2 minutes of inverting, we will be ready to put the lysate through the column. We have ready some of our materials that we'll need for this. We have the plunger already removed, and we have the barrel of the syringe screwed onto the wizard column, and then we can put that into an Eppendorf tube. So what we'll do next is we'll add this resin plus lysate into the column. I'll get my other syringe ready. I have my column ready. I'm going to pull out the plunger and attach the barrel to my column. I have these over top of sterile Eppendorf tubes. We're going to put the waste in the tube where the resin is going to come from. So the easiest way to do this is actually to use serological pipette. We should have 3 milliliters in this conical tube, our DNA resin, plus our lysate that has been nuclease treated. And we want to add 1.5 milliliters to each of those syringe barrels. So I'm just going to use the same pipette, and the serological pipette is easiest to do this, and add 1.5 milliliters to each of those barrels. Be careful to not knock these over, and keep that conical tube. That will actually be used for waste. So I'm going to open up the tube that we originally got our resin and lysate from, and I will take this syringe, put it over top, and take the plunger and put it inside. And you'll want to be careful with this. Hold on to the column as you're doing this so that you don't lose the column into the tube. And carefully push down on the plunger. And you'll see that there's a bit of resistance at the beginning, and then this should drip through. I'm going to put that back on a tube. I'm going to get my other one and filter that through. Again, hold onto the column, dripping that liquid into what will now be our waste container. Now that we have the DNA in the column, we want to wash that DNA. So what you'll want to do is actually unscrew the column from the rest of the syringe. This is important because if we pull up on that barrel with the plunger inside and to take the plunger out, which we need to do next, we'll actually disrupt this important filter that's inside the column. So don't forget to unscrew this before you take the plunger out. I'm going to do the same thing here. Unscrew this and take the plunger out. Now we can screw this back on. And like I said, make sure you just go to the point of resistance. And now we're going to wash our DNA and wash the column using isopropanol. We have a sterile work area, but it's good to use a paper towel to put these plungers on so that you're keeping the workspace more or less sterile and you're not going to pick up DNA from somewhere else or a chemical that could hurt that DNA. So now we're going to wash the column, and we're going to do this 3 times, 2 milliliters at a time. You can use the same serological pipette for this, and we will be using that 80% isopropanol. So you get 4 milliliters of isopropanol, 2 milliliters in the first column, 2 milliliters in the other column. And we will do just as we've done before. where we take this off, put this over our waste container, put on the plunger, and hold on to the column so that we don't lose the column, and then push through as it drips. Okay? We can take off the column so that we can pull out the plunger and then put the column back on. Now we can do this with the other one. Take off the column, take out the plunger, and reattach the barrel and the column to each other. And you are going to repeat this 3 times so that you've washed each of those columns with 6 milliliters total of isopropanol. Each time collecting that lysate into what was your DNA resin tube. This should fill up pretty much all the way. If you end up running out of room, let us know, but you should end up using all of the isopropanol provided. Once you've washed each of your columns 3 times, what we're going to want to do is actually get some new Eppendorf tubes, because the ones that we've been using were really just as a holder for our columns and may have some things that have gone through them. So we will get rid of those. So I'm going to take our column and I'm going to unscrew it from the barrel and get rid of that Eppendorf tube that was originally under there. So again, get rid of the barrel and use our new Eppendorf tube. So now we have our DNA in the column and we eventually need to elute it. But just to make sure that we get out all of the isopropanol, we're first going to centrifuge these columns. This is a fixed rotor centrifuge, and you'll want to make sure that your tubes, and you'll probably have to coordinate with other people in your class, are balanced. You can see that we put those 2 tubes opposite from one another within the centrifuge. Make sure you put on this lid. This will make sure that nothing comes flying out. Close the lid. We're going to spin it for 1 minute. And that, like I said, is just to get rid of any extra liquid and especially any residual isopropanol that's in the column. Press to start. If the centrifuge sounds strange and something is off balance, press stop immediately. It should just be a humming like this, which would indicate that things are running smoothly. After 1 minute, the centrifuge will actually stop on its own and the lid will open. And then you're going to pull this off and take out your tubes. We're going to discard the tubes that are here. You can see that there is some liquid that has collected into these tubes. We don't want that. Our DNA is still in the column. So we'll discard these tubes and get 2 new ones. This will be the last time we discard any tubes because these 2 tubes will have our DNA. So I'm going to take the column and just transfer it to those tubes. Immediately get rid of the old ones. So like I said, this is an important point because this is where we're actually going to elute our DNA. Make sure that you're sort of thinking about at this step that we're not getting rid of anything anymore. This is the important part of where we're going to take that DNA, which is bound to the column, and actually elute it into the. We will have a heat block set up in between the step of elution and the step of spinning down that isopropanol with a 90-degree heat block. You'll put it in there for about 60 seconds to just evaporate any of that residual isopropanol that could interfere with the integrity of DNA that you're eluting. To elute the DNA, there will also be tubes available for you that are going to be at 90 degrees. It will be sterile water, and you're going to use that sterile water and add this directly to the column. 50 microliters of water into each tube. It is good to switch tips in between here. So when you do this, I'm going to enter the pipette tip, and you'll want to kind of put this on an angle so you can see well into the barrel of the column. And I'm going to put this on an angle and pipette just over the filter. Do not pierce the filter with your pipette tip. So be really careful about this. You can look on the side to see where that pipette tip is, and you can look from the top and just make sure that you don't actually pierce the filter because that may damage the filter. Change your pipette tip. Get more water. This is the 90-degree hot water. And again, go to your other column. Make sure you know which one you've added the water to because it will not be obvious. That water will soak into the filter and you won't be able to see it. So now that we have water added to each of these, we want these to sit for 1 minute at room temperature, and then we're going to centrifuge to elute that DNA. After about 1 minute, and longer is fine too, we're going to take these tubes and put them back into the centrifuge to elute our DNA. So we're back at the centrifuge. Again, make sure things are balanced and they're across from one another. On the lid and start. 1 minute's up. Take out your tubes. You should have your liquid inside at the bottom of the tube, and the filter shouldn't have any DNA or liquid left in it. Everything should be collected now into that collecting Eppendorf tube. You'll want to make sure that you get rid of the filter, but you close up these tubes, and that's what we're going to hold on to. Those tubes now have your DNA in them. Label these tubes with your names, the names of your phage, and eventually also the concentration of DNA. And we'll do that using what's called a nanodrop. So at this point, it's probably best to actually combine the DNA from each of the tubes that you've eluted. So using your micropipetter, pipette out 50 microliters of DNA from one tube, get all of it, and pipette it into your other tube. So all 100 microliters of DNA should be in the same tube, and that's what we will read in the nanodrop. We'll hold on to that DNA and stick it in the fridge, and hopefully we will have enough to actually send for sequencing. We can't send DNA for everyone's phage. We'll only be able to send one per class, so we'll probably pick the phage that has been producing the highest titer, as well as the highest concentration of DNA. Next, now that we have the DNA, we'll move on and do restriction digests, which is taking these restriction enzymes and allowing them to digest that DNA that we've isolated and seeing what restriction patterns are produced when they've cut DNA at particular places. And we'll talk about that in the next video.